All right, now we're ready to go with the English. I hope everybody's ready because it might be a bit challenging. It's a bit fast and a bit technical. It will be a presentation from Magali Leroy. She's a microbiologist, so already that scares us a little bit, with extensive expertise. Oh, I can hear myself. Okay. I think we're good now. With extensive expertise in molecular biology and genomic analyses. Her extensive studies and research have taken her all over the world, as she holds several research awards, including Grand Challenges Explorations in Global Health Prize from the Bill and Melinda Gates Foundation, and also a Nestle Research Award. Her hobbies include the vulgarization of extremely complex subjects for laymen, and let's hope she puts that hobby to good use today, because her presentation will be on genome-wide approach to vaccine target candidates identification against otitis media causing non-typeable Haemophilus influenza. So good luck. I'll just see. And again, it's a pre-recorded. Oh, there we go. We'll see if it works and we're ready to go.
Okay, so I'm presenting today part of my thesis research project on vaccine target identification regarding specifically a non-typeable Haemophilus influenzae, which causes otitis media. So this work is 15 years old. So regarding technologies is not quite relevant today because of high throughput sequencing and new microarray technology that is now available. But nonetheless, it is the process through which you can identify candidate targets for the development of a vaccine. Okay, let me try to find how to switch slide. I'm stuck. Okay. So Haemophilus influenzae is a small bacteria that was identified a long time ago, and it's called influenzae because actually it was isolated from the lungs of people who had the flu. And originally they thought that was the pathogen responsible for the flu, but in fact it's not. It's a secondary infection that occurs in the lung often when people have the flu. And it's pathogenesis, so it's only host is humans, so it's only present in humans. And there are two types of Haemophilus. There are the typables and the non-typables. The typables cause a range of disease, including meningitis, arthritis, pneumonia, and other very severe disease, while the non-typable causes mostly pneumonia, otitis media, and bronchitis. The disease that we are specifically interested in for this study was otitis media, which is a middle ear infection in children. In 60% of cases, it is caused by non-typable Haemophilus influenzae. It is often associated with chronic disease in children, and that's a problem because that's the age at which they start learning language, and so having being partially deaf due to ear infection is actually not a good thing. And in developing countries, it's also responsible for 1 million deaths due to secondary pulmonary infections. So this is the reason that a vaccine was considered for development to curtail those diseases. So vaccine development, there was through history different strategy that were used. Initially, they used live attenuated pathogens. So often a sister disease that was less severe to inoculate people and to give them some immunity. We then moved to kill the whole organism. So we would use the entire bacteria and kill it and use it to inoculate people. The problem is that actually caused very often secondary problems, because if you have the entire cells, you have also all its content, including sometimes toxins. So the next step was to use purified protein or polysaccharides as targets for the vaccines. Also, that can include problems, but that was used for a very long time. And now we are moving to a new era, which is genetically engineered vaccines. So we can not just purify proteins, but we can actually synthesize specific targets that are of interest to generate a vaccine. So the good criteria for a target is that it needs to be expressed. So it needs to be present in the bacteria. it needs to be exposed, so on the surface of the bacteria in order to be accessible to antibodies. And it needs to be conserved to give a broad coverage. If you only have immunity against a specific strain, it's not going to protect you. You need to be protected against the entire potential population of the bacteria in order for the vaccine to be efficient. Regarding specifically HI and otitis media, there are a few vaccines that are already available. Against HI type B, which causes meningitis, there is a vaccine, but that type is not involved in otitis media. There is an heptavalent pneumococcal conjugate vaccine that exists that prevents otitis media against pneumococcals infections, but not otitis media. And more recently, there was a conjugated vaccine with a pneumococcal capsular antigen and an Haemophilus influenzae protein that was used. But as I mentioned before, if you want broad coverage, you want multiple targets and you want them to be conserved so that you have a broad coverage. So the goal of this study was to identify additional targets to develop a conjugated vaccine against non-typeable Haemophilus influenzae. So in the literature, you can find already potential targets that are surface exposed antigen of NTHi. But if you only base your research on the literature, this is the type of information you'll find. So the list here presents antigens, the surface exposed proteins. But as you can see in the far right columns, not all of them are considered to be conserved, so they could not be used for a vaccine. And even in those that are considered to be conserved, that are highlighted in yellow, you can see that if we check specific strains, actually they're absent in some strains. So they're considered to be conserved, but they're not so conserved after all. So in the lab I was working at the time, we had a very useful tool, which was what you can see on the left, a very wide collection of clinical isolates. So those were isolates that were collected in children in hospitals. And the phylogenetic tree that you see on the left was organized using RRFLP, which stands for ribosomal restriction fragment length polymorphism. So what we do is that there are ribosomal operons, there are 6 of them in HI, which we cut with restriction enzyme, and that generates a profile that we can then use like a fingerprint to differentiate between the isolate and determine how far away from each other they are genetically. What you can see on the tree on the left, so those are all the isolates organized by their genetic difference, their genetic variability. And to the right, you have a color coding, which shows the capsular type. So you can see, for example, that the red kind of cluster together. They are genetically very similar. But where you have no color, those are the non-typeable, so the one we are trying to target. And they are spread throughout the tree. That means they actually they encompass a lot of the diversity of the species. And that's going to be one of the difficulty of targeting this type or these non-typables, is that they're genetically very diverse. So the goal of this study was to identify new candidates, and for that we used 2 approaches to cover 3 of the main aspects of the targets that we want. First, we wanted to be conserved, and for that we had an in silico approach using already available sequence data. Remember, that was 15 years ago, so sequence data was much less available because high throughput sequencing was not something that was available and that was cheap at the time. The second aspect that we want is that the targets be present and expressed. And for that, we are going to be using an animal model and microarray analysis to verify that the targets are indeed expressed during the course of infection. The goal being to identify conserved and expressed candidates for the development of a vaccine. So the first step was that in silico analysis. So how do we identify the conserved genes, or at least conserved regions? So we downloaded all the publicly available sequences for non-typeable Haemophilus influenzae, which was then aligned, and we then identified the conserved regions. So a gene can be variable, but very often it has conserved regions that corresponds to constraint in the function of the genes. And using that, we created a genome-wide database, a database of the genome-wide conserved regions of Haemophilus influenzae. To test that database, we actually used a spotted microarray. That's what was available at the time. And we tested that microarray with what we thought was a representative set of the diversity of the species. So I'm going to zoom in. This is the tree, and you can see with the green arrows the isolates that we choose to cover the diversity of the species and to test those conserved regions that we had identified. Excuse me. So that spotted array contained 166 genes, 180 probes, because we had duplicates. They included non-typeable Haemophilus influenzae genes, including controls and other species like the chinchilla, since the model is going to be in the chinchilla. And what we found was a pretty good level of conservation experimentally. If you remove 9 genes that turned out to not be conserved after experimental verification, we had a probe consistency of 94.6%, which is pretty excellent and shows that we can verify experimentally that the conserved sequences, the conserved region that we identified in silico, were indeed pretty conserved within that subset of 8 isolates, which is supposed to encompass the diversity of the species. So the next step was to identify those targets that were expressed during the course of infection in an animal model. The animal model that we use is the chinchilla, from which we can then recover ex vivo samples, purify the RNA, and then use a microarray to see what is expressed during the course of infection. So typically, this is the types of sample we will recover. So you have a green and a blue line. The green line represents middle ear sample. It's a middle ear infection. So we would recover samples directly from the middle ear in the chinchilla. And we would also recover washes from the nose, the nasopharyngeal area, where the colonization occurs. The problem that we were facing at the time is that the maximum amount of samples that you can recover from a middle ear or from a wash of the nose is around 10 to the 6, 10 to the 7 cells, 10 to the 4 cells, sorry. For microarray, you need 1,000-fold more, so that's not enough. One of the options that we tested at the time was to do multiple washes. So what you're watching now is not over time, it's a single animal, a single day, and instead of collecting a simple fluid from the ear or a single lavage from the single wash from the nose, we did repetitive washes of the middle ear and repetitive washes of the nose to see if there was anything left in there to collect. And what you can see, this is one of the best examples, of course, is that the initial sample is not all that is in there. It's not all that's in the middle ear or all that is in the nose. And you can wash multiple times and recover the same amount of bacteria. So there are, if you do 10 washes, 10 times more bacteria in there than you initially thought. And for the nose, we could go up to 20 lavage, 20 washes, and still recover as many bacteria almost as initially. So that gives us a 10 to 20-fold factor. When you look at the washes through microscopy, you can see different things. So on the left, you have the inoculum, which is the lab culture that we inoculate to the animals. And on the right, you have the middle ear fluid that was collected from a chinchilla. The white arrows point to small bacterial agglomerates. The big cells or epithelial cells from the chinchilla, and the small round cells or red blood cells that were collected during the washes. What we see if we pull samples, so we pull the first 5 and the first 10, and we observe them through microscopy, is that you can observe biofilms that form in the middle ear. So the first lavage, you collect the mucus top layer of the biofilm, and in the last 5 lavage washes, you recover the matrix, which is the tightly packed bacterial cells that constitute the base of the biofilms. So the multiple lavage sampling actually was published and was very relevant because it first revealed that there was a higher load of bacteria in there that was initially thought, which is very important for antibiotic therapy. It also provides a mean to study biofilm in vivo more easily. It shows the different types of bacteria you have during the infection process, and filamentous types, for example, are important because those are highly resistant to antibiotic treatment. Biofilm encased bacteria as well. So all those are points that support the development of a vaccine. It also points to the fact that because there are biofilm that form very early on, you need to target with a vaccine very early on during the colonization and the infection process before the biofilm establishes. So we will need to find targets or express very early on during the colonization and infection process. So back to the microarray to see what targets could be expressed early on during the disease. And we're still short of 100-fold amount of bacteria that we need to do the microarray. So we resorted to RNA amplification. So the prokaryotic RNA that we collected in the samples that were purified do not have poly-A tails like in eukaryotes. So we did a reverse transcriptase to move on to cDNA, which is much more stable, to which we added poly-T tails. And then that we used a T7 promoter and poly-A tail linked. The T7 promoter is a high throughput promoter that allows to synthesize RNA in a very important amount. And we did that in 2 consecutive rounds to multiply the amount of RNA that we had. We had about 100 to 150-fold increase, which was enough to do the microarray. I'm not showing the microarray data here. It's way too complex. But what I'm showing here is the result of it. So what we are comparing in red is the inoculum that we used in the chinchillas. And then in blue, you have the day 5 middle ear samples. So what you can see is that the inoculum are tightly packed together. So the 2 rounds of amplification did not inherently create a bias or a variability. It could have introduced technical variability that we don't see. So that's a good point. It means the RNA amplification did not create variability. However, you can see the blue dots are kind of scattered around, and that shows the biological variability between animals and between samples. So how do we identify the targets? Because that was the goal. Now that we have the microarray analysis, we pulled all the genes that were expressed in all 3 day 5 middle ear samples. We compare that to our in silico analysis, so to the database of all the conserved regions. And then we also included the annotations, so all the genes that potentially could be expressed, so accessible to the immune system, so cell wall, envelope membrane, transport protein, and all those that were even just predicted as potentially encoding surface proteins. And that gave us 15 candidates that were expressed, potentially accessible on the surface of NTHi. Those are the 15, and I'm going to point out, I should have pointed out earlier, but so those are the 15 candidates that we identified through this study. In summary, so this study of genome-wide screening for target vaccine identification used in silico analysis and a database of all conserved regions that we generated, a multiple lavage to increase the amount of samples, and also that gave us clinical insight into the colonization and infection process in the chinchilla model. We had to use RNA amplification because we did not have enough ex vivo samples for the microarray, but it turned out to be efficient enough to allow us to study the ex vivo gene expression with microarray. That gave us 15 candidate targets, 6 that are more promising based on their annotation as being surface exposed. But we know that these 15 and potentially more promising 6 are conserved, expressed early, so at least at day 5 in the nose and or in the middle ear, and are potentially exposed on the surface and so accessible to the immune system. Thank you very much.
Hope that wasn't too hard. As you can see, if you look at the chat, thank you so much to our interpreters in the booths. You guys were absolutely exceptional. We will now take a break for about 20 minutes so that you can do your feedback. And we will come back after that to discuss how everything went, including ranting about how hard this was. So thank you again to everybody, and see you all in about 20 minutes. We will have a slide up with specific time to come back. Thank you so very much. Bye.